Hello viewers, in my previous lecture I had explained you various stages of menstrual cycle as well as the process of oogenesis which results in the formation of the secondary oocyte or ovum. Now here you can see I have today I will be beginning with the transport of ovum and the process of fertilization and the passage of growing embryo through the fallopian tube. Now this second meiotic division, and I had already discussed with you that the first meiotic division is completed before ovulation, and now the second meiotic division, now spermatozoa, it enters the zona pellucida layer and the plasma membrane or the vitelline membrane, and it reaches the ovum. This induces the completion of the second meiotic division. It results in the formation of an haploid ovum or ooted and a second polar body. Now this ooted or the haploid ovum, now it is ready for fertilization. Then I told you the spermatozoa enters or strikes the ovum wall, that is zona pellucida, and the reaches from the plasma membrane, tries to gain entry inside for the process of the fusion. Now I told you it also takes when it takes the entry from the animal pole, it's very easy to get fertilized, and its nucleus get fertilized with the nucleus of the ovum, and therefore I told you the egg which is released here outside the ovary, it is non-motile and it is picked up by these fimbriae which is the part of the fallopian tube, and these fimbriae takes up the ovum, and by the ciliary movement, here it is the ciliary movement, many cilia are located in these finger-like projections called as fimbriae, and it takes the ovum inside, and this ovum reaches by the muscular movement, it reaches the fallopian tube, and here it meets the spermatozoa which is travelled from the vaginal tract and reaches here at the ampulla. The process of fertilization takes place at the ampulla, or you can say here ampullary stomach junction. Then soon after the fertilization there is formation of a zygote. The ovum is haploid, sperm is haploid, and the formation of the new cell is zygote, which is now a diploid cell or the 2N cell. The sex of this zygote is decided here only, at this stage only. Now the female is having X chromosome and the male is having X and Y both. That the sex chromosome of the female is X only, and when the X of the female fuses with X of the male, because the male carries 50% of spermatozoa having X chromosome and 50% of spermatozoa having Y chromosome. So when X of the female fuses with the X of the male, then here the fetus or the zygote, it will be a female, and when X of the female fuses with the Y of the male, then the fetus here will be a male child. So the sex of the child is also decided at this moment only, and later on this zygote undergoes the process of divisions, many mitotic division. This process is termed as cleavage. This zygote undergoes many mitotic division to form an embryo, and this is termed as cleavage. Cleavage occurs within 30 minutes after the fertilization. I am repeating, cleavage occurs within 30 minutes of the fertilization. The process of cleavage is holoblastic in human beings, that is holoblastic, because I told you already that the human egg is allocetal egg. It lacks yolk, or it contains negligible amount of yolk. Okay, so here you see that the zygote, the process of cleavage is holoblastic, and it is meridionally vertical. Meridionally vertical. That means it is dividing from animal pole to vegetal pole. I am repeating this. The cleavage is holoblastic, that is, it is meridionally vertical. That means the division is from animal pole to vegetal pole, and therefore formation of two daughter cells, here you can see formation of two daughter cells. Now the second cleavage, it will also be meridionally at right angles to the first one. Second cleavage will be meridionally at right angles to the first one, and formation of a dumbbell shape. Here you can see the formation of dumbbell shape. Therefore, in the process of the cleavage, formation of the cells, you say it will be like that: 2, 4, 8, 16, 32. So this I mentioned to you, the second cleavage, it occurs at right angles to the first and formation of a dumbbell shape. Then subsequently many cleavage occurs one by one and they are in a proper manner. All the subsequent cleavages further will be taking place and they will be in a orderly manner, and therefore formation of a solid ball of cells, that is, I told you here, formation of 2 cells, then 4 cells, then 8 and 16 and then 32. Then 16 to 32, it is, the stage is called as morula, because here the blastomeres, they become smaller. Blastomeres goes on becoming smaller, you can see. Whereas you can see the rest, the size, volume and the shape, it is all similar throughout the process of cleavage, because there is no growth in the occurring in the blastomeres. So they go on becoming smaller and there is no change. That means the size, volume and the shape is similar throughout the process of cleavage. So here this solid ball of cells, it is 16 to 32, it is called as morula and looks like a mulberry. The botanical name of mulberry is Morus alba. Therefore, the name of this stage is morula, because at this stage the solid ball of cells looks like mulberry. This morula slowly and slowly, it's not very slow, soon it gets transformed into blastocyst. In this stage, what happens? There is formation of an outer layer and the aggregate mass of cells to one place, to a one particular place, and there is increase in the cavity or the formation of a cavity. This cavity, it is called as blastocele. This cavity is called as blastocele and it is filled with a fluid. At this stage, this blastula, or you can say it is termed as blastocyst, or it is called as blastodermic vesicle. It is also called as blastodermic vesicle because of the formation of a cavity or development of a cavity, and this cavity is filled with a fluid called as it is called as blastocele fluid, blastocelic fluid, and this stage that is called as blastodermic vesicle or blastocyst. At this stage, this embryo gets embedded into the, or it gets fixed to the endometrium lining of the uterus. I am repeating, at this stage, that is blastocyst gets fixed to the uterine lining of the female or endometrium of the female, and this process is termed as implantation. It takes place around about 7 to 10 days of the fertilization and it goes on developing, and I told you it is having this outer wall which is made up of a line of cells, and these are termed as trophoectoderm or trophoblast, and these are the inner mass cells. Aggregate of cells are also formed here which are called as inner mass cells. These, the trophoblast term has been derived from the Greek word called as trophos, means to feed, to provide nutrition. So this trophoectoderm, it is made up of inactive cells, and these cells only provide the nutrition and helps in the protection, whereas the inner mass cells later on gets differentiated in the formation of the embryo. So this way this trophoectoderm or the trophoblast provides the nutrition and also help in the formation of many membranes for the deciduas, and these are giving rise to later on into many extra embryonical membranes. They are chorion, allantois, yolk is negligible, and the chorionic will chorion will help in the formation of the villi sort of structure, which will help in the formation of the placenta. So it will give rise to embryonical membranes, that is chorion, allantois, yolk and the amnion. So this way, this will amnion membrane will form the amniotic cavity. It will protect it and it will also help in the desiccation and means it will not allow it to desiccate, whereas the chorionic and allantois help in the formation of the placenta, and then it will be attached to the uterine wall. It gets interdigitate with the tissue of the uterine lining, and it will help in the formation of the placenta. Therefore, it will help in providing the nutrition, whereas the innermost cells get later on differentiated into the embryo. This way, the whole development will take place here in the uterine lining or the endometrium. Then I told you already that this size and the volume and the shape is similar all throughout the process of cleavage. Now, whereas here the blastomeres, which were called as two, which are in the numbering is like that: the first formation of the 2 cells, then the 4 cells, then the 8 cells, and then 16, and then the 32. So 16 to 32 stage, I told you, it is termed as morula, which is a solid ball of cells, looks like mulberry, and it is termed as morula because mulberry's botanical name is Morus alba. So since at this stage it looks like mulberry, mass of cells looks like mulberry, small ball, and therefore its name is morula, which later on gets transformed into the blastula, or the formation of blastocyst takes place because of the formation of a cavity in the center, and this cavity is termed as blastocele, and the occurrence of the innermost cells at one side and the line of cells at the outer layer. This outer layer, it is the round outer layer cells called as trophoectoderm or trophoblast, and the innermost cells. This way, at this stage, the blastocyst gets fixes to the uterine lining or the endometrium, and we say the female is pregnant, and this is the process of implantation which takes place around about 7 to 10 days after the fertilization. Thank you.